Welcome, everyone. My name is Mahafuzur Rahman. I'm a PhD student at the Department of Chemistry and Biotechnology. The title of my talk is Binding of Human Proteins to Amyloid Beta Protofibrils. This is a graphical representation, graphical abstract of my talk. Here you see this is the binding of human proteins to amyloid beta protofibrils. The core protein here, this is a hexameric model of amyloid beta, and we have developed this model in our group. And the proteins surrounding here, those are the different proteins from our biological fluid. But before going to show you the results, I would like to give you a very short overview of what is Alzheimer's disease and how it is related to these amyloid beta protofibrils. Well, Alzheimer's disease, this is a progressive neurodegenerative disorder that attacks your brain nerve cells. And the results are you are losing your memory, intellectuality, thinking capacity, and some other behavioral changes as well. The target group for Alzheimer's disease is 65 years or older. Although the risk of having Alzheimer's disease increases with the age, but Alzheimer's disease is not a part of normal aging. This is a disease. So, what is the difference between a healthy brain and an Alzheimer's brain? If you, sorry. This is the view of a healthy neuron, and this is the view of Alzheimer's neuron. In Alzheimer's brain, two abnormal protein structure. One is called the neurofibrillary tangles, which is a two-state fiber of tau proteins that form a hard, insoluble structure inside the nerve cell here. And the other one is called beta amyloid plaque. This is mainly composed of amyloid beta, and that forms a hard, insoluble structure outside of the neuron cells, between the neuron cells. In our group, we are interested with the amyloid plaques. More specifically, we are interested with the proteins that is the major component of these amyloid plaques, that is the amyloid beta peptide. Amyloid beta peptide is a cleavage product of amyloid precursor protein, APP. Amyloid precursor protein first cut by a beta secretase, and then the N-terminal domain subsequently cut by gamma secretase, and that gives a peptide that's called amyloid beta peptide, which is a 43 amino acid residues long. And up to this production, it's happening in the normal brain and in Alzheimer's brain as well. But the difference is, in the normal brain, these peptides are usually broken down and eliminated, whereas in the AD brain, these peptides undergo some conformational changes, and then it's deposited as this plaque in the brain. If we look at the A-beta fibrillation pathway, you see here this is the unfolded monomer, and that goes to this oligomer or protofibrils, and subsequently the end product is the fibril aggregates, and that is the major component of the amyloid plaque. Here, the intermediate species that's called oligomer or protofibrils, this species seems to be neurotoxic, and it's caused neurodegeneration in the Alzheimer's disease. But the mechanism by which this species become neurotoxic is not known. But an important hypothesis now is these intermediate species may have some interaction with other proteins from our body fluid, and that makes this species neurotoxic, and it's also kill the neuron cell in Alzheimer's disease. To test these possible interactions of these A-beta protofibrils with other protein from our body fluid, we have developed a pull-down assay where we used a tosyl-activated magnetic bit to couple A-beta on it, and then we have incubated this A-beta bit whole complex with human samples, human body fluid. We have used serum sample and CSF samples. We have used 1 serum and 12 CSF samples. From these 12 CSF samples, we have tested 6 Alzheimer samples, and 6 healthy samples. And followed by incubation, we have eluted the proteins and then separated on the SDS space. And then to reveal the identity of the proteins, we have done mass spectrometric analysis. And then a subset of the proteins were evaluated using surface plasmon resonance technology to know how strong these proteins are binding to each other. And this is the SDS space images of human protein binding to A-beta protofibrils. As you have seen, there are several clear bands for the protofibril samples. Whereas binding to the controls, this is a glycine-coded bit. Sometimes this is a tryptophan-coded bit. Binding to the control is very less. And also binding is less to the monomeric beta. What we have done then, we have cut this whole gel lane, except the regions here, this is corresponds to A-beta monomer, dimer, and trimer. We have cut this whole gel lane, and then we proceed for mass spectrometric analysis. And through the mass spectrometric analysis, we have identified a substantial number of proteins that binds or come up with amyloid beta protofibrils. And according to their biological functions, the identified proteins that could be distinguished in few main biological groups. We have proteins that involve in lipid metabolism and transportation. We have proteins that is complement-related inflammatory proteins. We have proteins that's involved in hemostasis. And interestingly, all of these groups are somehow correlated in the Alzheimer's biology. For example, amyloid beta has shown active the complement pathway in the absence of immunoglobulin bindings. And this is a very common case for neurodegeneration diseases. And lipids, those are always close to A-beta and Alzheimer's disease as well. Most interestingly, this apolipoprotein E and apolipoprotein J, these two proteins have been discussed to play a crucial role in Alzheimer's disease. And we have identified several proteins here from the apolipo group. And you can see some proteins with this asterisk. These proteins are reported to be present in the amyloid plaque. This is an indication that our study is also relevant in vivo condition. These are the SPR data of the 4 selected proteins. And here is the circle lines here. This represents the raw data, and the black one is the fit. And we have find that all of these 4 tested proteins that bind to A-beta very strongly with low micromolar affinity. It binds with 0.3, 0.6 micromolar affinity, 3 micromolar affinity. And interesting is here, what interesting is that apolipoprotein E, which is the only one genetic risk factor for Alzheimer's disease, which binds with most strongly to amyloid beta protofibrils with just 3 nanomolar binding affinity. And here we conclude our study. Amyloid beta protofibrils, it has the ability to interact several proteins from human serum and cerebrospinal fluid. And we also conclude that protein aggregation that enhances protein binding, as we have seen a very less binding to monomeric A-beta. And we also hypothesize that protein ligands should be considered in any study that explains the neurotoxicity of A-beta that's specifically linked to A-beta aggregations. And I would like to thank you for listening to me.